Okay, so yesterday we ended on looking at isomers. We defined isomers as compounds or organic molecules having the same molecular formula but different structures, and therefore they have different properties. And then we went on to classify, and we said that there are basically three types of isomers. So we said there are structural isomers, geometric isomers, stereoisomers. Then we went on to look at the structural isomers. We said that these differ in their covalent arrangements of the atoms, and we gave examples of glucose and fructose as structural isomers. Geometric isomers, we said they're identical in the arrangement of their covalent bonds, but differ in the spatial arrangement of atoms or groups of atoms. So we talked about the cis and trans isomers. They said that when it's designated as cis, simply means on the same side. It simply indicates that the two large components are on the same side of the double bond. If it is trans, it means that they are at the opposite or on the opposite sides of the double bond. So we're going to find the cis and trans, especially when we start looking at the lipids. Then we have another form of isomers or isomerism, where what we call stereoisomerism. So under stereoisomers, they have the same carbon as skeleton, but differ in how the groups attach to this skeleton as they are arranged in space. So the stereoisomers contain the same atoms that are bonded identically, but the bonded atoms are oriented differently in space. That is to say that they have identical bonding constitutions, but they differ in how the atoms are oriented in space around the atoms to which they are bonded. So when you look at stereoisomers, they can further be subcategorized into diastereomers and enantiomers. So they're of importance for discussion are enantiomers. So enantiomers are more like mirror images of each other. So for example, if you can see in the pictures, these are mirror images of each other. So you find that when a carbon is bonded to 4 different groups, the molecule that is formed is what is called the chiral molecule. So it's chiral molecule. So a chiral molecule have stereoisomers that are mirror images. So when you look at that image, those 2 molecules have the same 4 groups, but they cannot be superimposed imposed on each other. So just like our hands, we cannot superimpose them. For them to be superimposed, we would need to flip them. So these are what we call the enantiomers. So when you talk about chirality or a chiral molecule, we are simply referring to handedness of a molecule. So chiral molecules have a chiral center, and this pair of molecules cannot be superimposed, as we have already said. So a chiral center is an atom which has 4 different atoms that are bonded to it. So it has 4 different constituents that are bonded to it. So when you look at a carbon atom, when a carbon atom has 4 different groups that are bonded to it, it is called asymmetric or a chiral carbon. It is called asymmetric or a chiral carbon. So chiral compounds are characterized by their effect on polarized light. So we have those that can rotate to the right. These are what are called dextro. or refer to dextro, and if they rotate to the left, it's called the levo. So two chiral forms that we have is D for dextro rotatory and L for levo rotatory. So we'll find that we have optical isomers such as D-glucose. We're going to find that there's D-glucose. We're going to find that there's also L-glucose optical isomers, since they are polarized light. So we have D-glucose and L-glucose. So when you look at the enantiomers, they are identical in most physical and chemical properties. So find that they are identical in the melting point or in the boiling point, or density and chemical reactions are typical for the functional groups that are present in the molecule. However, there are certain differences that can be discerned. So as we have already mentioned, we find that they differ in the way they polarize the light. And also, they also differ in their interaction with other chiral compounds. So the chiral molecules specifically recognize other chiral molecules. So for example, if we are to look at the L amino acid protein enzyme, they are able to recognize other chiral molecules. So when you look at the living system or biological system, they tend to produce only a single enantiomer of the two possible forms. So for example, mostly in our systems or in the biological systems, we find primarily D sugars and L amino acids. So most of the sugars are D sugars and the amino acids are L amino acids. And the other part that we need to look at is biological molecules as polymers. Biological molecules as polymers. So when we look at the term a polymer, it is a long molecule that is built by linking together a large number of small and similar chemical subunits that we call monomers. So a polymer is made up of small subunits that are called monomers. So we find that biological molecules, for example, proteins or nucleic acids, they are very large and they consist of 1,000 of molecules or atoms, 1,000 of monomers. So these are what we call the macro molecule. So the word macro meaning big or large. So some of the biological molecules that we have carbohydrates. So we do have complex carbohydrates, for example, starch, glycogen, which are composed of simple ring-shaped sugars. So I find that the nature of a polymer will be determined by the monomers that is used to build that polymer. Then we also have nucleic acids, which we've already looked at. And we said that nucleic acids are polymers of nucleotides. And then we have proteins, which are polymers of amino acids. So those 20 amino acids can be linked in an endless way to form proteins. And then the other polymers that we have are the lipids. So lipids are polymers of fat acids and glycerol. Fat acids and glycerol. So polymers that we have carbohydrates, nucleic acids, proteins, and lipids. Now, how are these polymers broken down or built? So when you look at these molecules or the polymers, they are built via chemical reactions that we call dehydration reactions or condensation reactions. And then to break them down, they undergo a process that we call hydrolysis. Hydrolysis. Okay, so a dehydration reaction. So for two monomers to form, for two monomers to form. So there on the dehydration reactions, you have one monomer and another monomer undergoing a condensation reaction in the presence of an enzyme to form a dimer. So for that covalent bond to be formed between two monomers, an OH group is removed from one monomer. An OH group, a hydroxyl group, OH group is removed from a monomer. And then a hydrogen atom is removed from the other monomer. So we have OH being removed from one monomer and an H removed from another monomer, which is going to lead to the production of that water molecule. So a condensation reaction will result in the production of a water molecule that we see there. So when we look at this simple chemistry, it is the same that we find in the linking of amino acids together to make a protein, or when we are looking at assembling of glucose units to make a polysaccharide such as starch. This same reaction is also used to link fat acids to glycerol when we start looking at lipids. So it's this chemical reaction is called condensation or dehydration reaction because of the removal of the OH and H, which is the same as the removal of a water molecule that is given out as we have seen. So this is a condensation reaction. So these polymers are made via the condensation reaction. And then they are degraded via hydrolysis, a hydrolysis reaction. So when we look at the cells, they disassemble macromolecules into their constituent subunits through this hydrolysis reaction, and a molecule of water is added. So under hydrolysis, we see a molecule of water being added instead of being removed. So this is a process that we call hydrolysis. So in this reaction, find that a hydrogen atom is attached to one subunit. So you have a hydrogen atom being attached to one subunit or one monomer. And then we have a hydroxyl group attached to the other monomer, breaking a specific covalent bond in the macromolecule. So this covalent bond has been broken down. And then we have a hydrogen atom added to one monomer and a hydroxyl group or OH added to the other monomer. So this leads to the breaking of the macromolecule. So this is a table showing the macromolecule, the subunit, the function, and the example of things that contain such a macromolecule. So for example, under carbohydrates, we have polysaccharides such as starch, glycogen, which are basically made of monomers of glucose. We also have cellulose, which is made of glucose as well. Then we have chitin, which is made of modified glucose. So when we look at glycogen and starch, these are storage molecules. They are energy storage molecules. So starch in plants and glycogen in animals. And then when we look at cellulose, this is a structural support. It offers structural support in plant cell walls. And when you look at chitin, it's for structural support and is found in exoskeletons of certain organisms, such as the arthropods. Then we have nucleic acids, which we have RNA and DNA, which are genetic or carriers of genetic information. Then we have proteins, which are made of subunits of amino acids. And the proteins could be used for as catalyst, as transport, and also for support as well. And then we have lipids, such as the fats, the phospholipids. So the fats could be used for energy storage, The phospholipids are constituents of cell membranes. Then we have other types of lipids. So from this, we see that macromolecules are actually made of subunit or monomers, and then they are broken down. They can be broken down to produce those same monomers. So you need to know the monomers which produce certain, these are macromolecules. You need to know the monomers producing these macromolecules. And then you also need to know the examples of the isomers, the structural isomers, optical isomers, the stereo isomers. And then you also need to know the functional groups, which functional groups are found in carbohydrates, which functional groups are found in nucleic acids, which functional groups are found in proteins, and which functional groups are found in lipids. Do we have any questions before we shift to another topic?
Yes, madam. Okay.
Okay.
Also clear.
Okay, thank you.